So these next two chapters have a lot of technical names that you need to remember in terms of terminology. This chapter here on synapse, pretty much dealing with chemicals. So the first part of the chapter, do not get too lost in the topics, but how is it that we even think about chemicals having an influence on the brain? Many of the studies came from looking at heart studies and taking the heart of an animal and the chemicals that are flowing in it. You take that same solution and put a heart in there from another animal, and that heart responds to the chemicals from that other heart. So there must be something going on with chemicals flowing, impacting behavior. So the synapse, again, is that space in between neurons. We won't go into the history. You can read that on your own in terms of who and how this was discovered and uncovered. Some terms to explain from the body to the brain. The synapses are the places in between the neurons that cause the connections to even work. So you get a synapse here, synapse here, synapse here, synapse here. So all those places where one neuron talks to another neuron is the synapse. In between there is where the chemicals are flowing. How this can be measured? There's something called a ESP, excitatory postsynaptic potential. Excitatory, that means stimulating. Postsynaptic means on the one that's being measured. Post. Remember we talked about in chapter 1, pre-synaptic terminal, post-synaptic terminal. Well, this is the post-synaptic terminal's potential being measured. You could also have an inhibitory postsynaptic potential, IPSP. So again, it's still the postsynaptic neuron being looked at and measured, but it's not being excitatory, it's being inhibited. So in the midst of the slides that are explaining this, know this one in particular. So we said in chapter 1 that at rest, the membrane potential is negative 70 millivolts. The bottom line here. If something happens like an action potential, that's where you get this large rise in the impulse. That's an action potential. Look at the figure. There's a dotted line right here. The dotted line is the threshold point. Threshold is the place in the location where there's going to be a firing of that axon if the impulse crosses that threshold. Like, unless you leave the room that you're in, you didn't cross the doorway or the threshold. So unless you cross that, nothing happens. So here, guess what? There's a little bump right here. Another little bump right here. The terms that were expressed at the beginning of that chapter, temporal summation, it talks about how many on top of one another could actually increase you to have an action potential. So here's a little bump. That's that EPSP we talked about. It's excitatory, but it didn't cause what? An action potential. Here's now two ESPs on top of one another, but they didn't cause the action potential. Here, they actually summed up enough to now exceed the threshold, action potential. Comes back to rest, another one happens. But guess what? It can also go below the resting membrane potential. And going below it would then be your IPSP, which we just talked about. So at the beginning of the chapter, as long as you know that particular figure, you're pretty good. So to move forward, to get through these technical terms, understand this. You can go ahead and read up on your own on that heart experiment I was telling you about, but that's how we kind of looked at chemicals from one place in the body can impact another place in the body, especially if we transfer it to other animals. So, for review, electrical chemical communication is how our bodies operate. Electrical is the electricity part, is the ions and how they work. The chemistry is the way the neurotransmitters are being emitted. Neurotransmitters. In between all that is a synapse. Between a presynaptic and a postsynaptic terminal, the synapse is where the chemical activity is occurring that we're talking about right here. Again, you read it on your own. If you look at the structure of trying to explain this, here's the axon. At the end of it is the presynaptic terminal. The synapse right there in blue. And then you have another postsynaptic terminal on the outside. This big green thing here is just saying it's a glial cell. Hey, it could be an astrocyte, a supportive type cell holding these neurons in place so they can do their job. So, axon, presynaptic terminal, synapse, postsynaptic terminal, these little blue areas here called receptors. A neurotransmitter chemical cannot work without a receptor. It's like a lock and key. So the key, neurotransmitter, must find the lock, receptor. Something to happen. So the chemicals are being released. I said before, I think chapter 1, about these little circles here are called vesicles. They're like containers. Inside the vesicles are the neurotransmitters. They're released across the synapse. We'll talk to the receptor. If it's the wrong receptor or the wrong chemical, no activity, nothing happens. You can read that on your own about how that operates. But let's go continue to further discuss this. Some of those neurotransmitters, you can know the labels of these as some could be amino acids, some could be modified amino acids, monoamines, neuropeptides, purines, and gases. Quickly, amino acids. Amino acids are the building blocks of proteins. Without amino acids, like Lego blocks, you don't have a structure. So if you put multiple amino acids together, you get structures. Small chain amino acids are called peptides. Large chain amino acids are called proteins. But those amino acids, without them from your diet, you don't consume the right foods, you're not going to have what your brain needs. So individually, some of them can function as neurotransmitters on their own. So glutamate, excitatory. GABA stands for gamma aminobutyric acid. Typical. GABA is inhibitory. Glycine inhibitory. Aspartate and others. Glycine, these amino acids, tyrosine could be an amino acid that could serve as a neurotransmitter. Phenylalanine could also be an amino acid serving as a neurotransmitter. And you'll see the structures of some of the chemicals how they're made in another slide. But again, first off, amino acids could be neurotransmitters. Secondly, a modified amino acid is acetylcholine. You'll see the structure how it can be made. I think it's acetylcoenzyme. Coenzyme A and choline put together make your acetylcholine. Well, choline comes from the diet too. Eggs, cauliflower, certain food items, they contain choline, and that is important for, again, movement and also brain thinking, like memory. Monoamines, amines. Let's see. Amine. Amine is like an NH2, nitrogen and 2 hydrogens on a chemical structure. Methyl groups, carboxyl groups, all those chemistry terms you may have heard of, those chemistry terms pertain to parts of the structure. So this mono one amine. These pretty much mean the same thing. Monoamine and catecholamine. Catechol, what does that refer to? So the names that you hear of some of these chemicals really pertain to the structure of what they have in them. When you start getting to some of the names of some neurotransmitters, there are names that are provided that may not necessarily talk about the chemical structure, but monoamine, you see in the book, is the same thing as catecholamine. Catechol nucleus is a nucleus, a major part of the structure that is a catechol. It's pretty much a benzene ring. So that's the catechol nucleus of the structure, catechol, and then you may have CH2 and an NH2. So the chemical structure, catechol nucleus, monoamine, these become very important because you ever heard of dopamine, norepinephrine, epinephrine? They all have that basic structure I just drew for you as their defining structure as to why they are that chemical. Neuropeptides, again, amino acids connect together, and some of them be endorphins, substance P, melanocyte stimulating hormone, neuropeptide Y, oxytocin, vasopressin, these small chain amino acids, like maybe 9, 10 linked amino acids, can be called neuropeptides. They alone in themselves can have neurotransmitter-like functions. And then purines, things like ATP, when you think of adenosine triphosphate or adenosine as neurotransmitters, they can function that way. And what's been found out in the last few decades is that there's some gases, like nitric oxide, they can also function as a neurotransmitter. So you can look here, it shows you again what I try to describe to you about catecholamines, also means monoamines. Here you have some places where some of the items or food items come from. This should be important to remember because it gives you a sense of, again, how each one of these particular chemicals is being made. Acetylcholine, as I said earlier, choline plus acetyl coenzyme A makes acetylcholine, a totally different structure than these. Here's your catechol nucleus right here, and the different chemicals that come off of it, or chemical structures that come off of it. So acetyl coenzyme plus choline makes acetylcholine. Right here, phenylalanine, amino acid from your diet. Tyrosine, amino acid from your diet. You don't get those, it doesn't make dopa, it doesn't make the dopamine, your norepinephrine, and epinephrine. You heard of serotonin, that functions differently. It's an indolamine, not a catecholamine. Tryptophan from the diet makes your 5-hydroxytryptophan, and then makes 5-hydroxytryptamine, which is then serotonin. That also then leads to making melatonin. So everywhere you see those arrows, enzymes. Enzymes are catalytic substances. They help to convert the chemical from one form to another form. So where those arrows are, if specific enzymes are missing or lacking, you're going to have a problem with chemical formation or neurotransmitter development. So we already said this, the neurotransmitters are stored in the vesicles. Once they're released into that synapse, they can't stay forever. So there are two ways we can speak of how they could be relieved or dismissed or gotten rid of or deactivated. One is called enzyme deactivation. One is called reuptake. When we say enzyme deactivation, we're saying there's chemicals that can be within the synapse that could wipe the neurotransmitter out in a way so it's not there anymore. One particular chemical could be monoamine oxidase. So when I said what those arrows are, what enzymes are, also remember that if you see a word with ASE on the end, pretty much enzyme. So monoamine oxidase, catechol-methyltransferase, that is COMT. So MAO and COMT are both chemicals that can break down your neurotransmitters and get rid of them so they're not just inundating your system. So the other one was reuptake, but we have a slide that will show that. I'll come right back to reuptake. But on this slide, it talks about exocytosis. Remember what that word means. It means the relieving of the chemicals out of a cell. First, a release of neurotransmitters from the presynaptic terminal into the synapse. So exocytosis, exo, release. If it says endo, it's coming in. And when we went over that information in chapter 1 about the nerve impulse, it's triggered by the active potential. So unless you have that electricity, there's going to be no chemistry, no chemical transmission. So there's some effects of the drugs or the chemicals, the neurotransmitters that can be ionotropic. Don't make it too complex. Ion. That means the ion is causing it to happen. Occurs when a neurotransmitter attaches to a receptor and immediately opens an ion channel. Could be that sodium we talked about earlier. Could be that chlorine we talked about earlier, or potassium. So ionotropic is when those particular elements are being impacted. Beyond ionotropic, it's also metabotropic. Metabolism. So it's going a little bit longer. Not as quick, fast acting as ionotropic. So it occurs when a receptor is initiated and there's a longer sequence of events that happens inside the cell. Remember from biology classes, like G proteins, there's other things that happen to change the cellular structure of that cell. So, metabotropic, neurotransmitter can talk to the receptor. It then causes these releases of things. Here's outside the cell, here's inside the cell. So it releases these G proteins and initiates second messenger systems, causing again the cell and the DNA and the gene to potentially change inside. So that's more longer acting. So we note ionotropic, quick acting, metabotropic, fast acting. And again, these are some of those elements inside the cell causing the longer change, again, changing the DNA in the cell, G protein. So think about stress that could happen and make your cells internally change. Neuropeptides, again, they can function as neuropeptides, small impact, or they can have a dramatic impact on the entire brain. They could have they modulate things. They don't maybe serve as a sole function by themselves, but they help to enhance other chemicals' function. Here's some distinctive features. You can look at that about what neuropeptides can function as. So in the chapter, it talks about drugs. And so I don't want to go too deeply involved in here until we have a discussion, maybe for one of our discussions on our Canvas platform. But you have stimulant drugs, depressants, opiates, hallucinogenics, drugs. Alcohol is a drug. These particular drugs affect different systems in the body. So hallucinogenic drugs affect things like serotonin. LSD is a hallucinogenic drug. LSD stands for lysergic acid diethylamide. LSD can send you on trips. And maybe you can fly through walls, jump off buildings. Well, Serotonin is a chemical that looks very like, well, LSD looks very much like serotonin. Serotonin helps out with your mood. Any imbalance in serotonin can make you feel depressed. Also affects something called obsessive compulsive disorder. Serotonin also makes melatonin. So this issue of mood and influencing your body's natural abilities to be sensing things like in a dream state, LSD can impact you and make you hallucinate, and it impacts some of your natural mechanisms. Nicotine. Nicotine is a stimulant. It says it affects acetylcholine receptors. I said earlier that acetylcholine can be important for movement and also help out with memory. So people who smoke cigarettes, maybe helps them to feel relaxed and recall things. Opiate drugs are drugs that can be used to calm you down and slow you down. So heroin, morphine, these are drugs that can be considered opiates. They all come from opium. Opium, that's right, that's stuff that's over in Afghanistan and people are fighting over it. Opium from the poppy seed. You ever had poppy seed muffins? Or poppy seed, bagels? So the poppy seed itself, people get out of poppy seeds. So the papaver somniferum, that's the chemical element that's in the poppy that makes this opiate give you that little high. So you now have this addiction now with adult opiates that are more impactful, like oxycodone, Oxycontin, all these particular pain medicines people are making and selling and getting addicted on. It's a problem today. So when you see that word endorphins, endorphins just means endogenous morphine. Your body's endogenous morphine. So you can get high on your own self. So earlier I said that you have the enzyme deactivation. You also have something called reuptake. So from the presynaptic terminal, postsynaptic terminal, when the chemicals are being released out of the vesicle, go talk to a receptor, it can't stay in there the whole time, so it can be reuptake, put back into the system. So that's reuptake, to put back in for use. Enzyme deactivation, MAO, would exist here to help come in and erase. Enzyme would just erase it here. Reuptake is it going back to the system. So those are two different ways that you can look at how the enzyme or the chemicals that won't be like continuing on to have an impact. And I mentioned earlier COMT, catechol-O-methyltransferase. That's what that stands for. Stimulant drugs. We did talk about nicotine, but you got amphetamine, cocaine. They help to stimulate dopamine. So dopamine, you know, is a chemical related to having euphoric responses, making you feel good. It's dopamine. Ritalin can also be looked at as a stimulant. So Ritalin is used for, attention hyperactivity deficit disorder. So a child who's always on the go, for some reason Ritalin helps slow them down to attend to things. So it's surprising how a drug that blocks the reuptake of dopamine can do that. The dopamine is called stimulation, but it's helping the person to attend at things. So Adderall is another drug for ADHD, but that's usually used for adults. It has a different little mechanism than Ritalin does. Cannabinoids, that relates to marijuana, that relates to the chemical substance THC, tetrahydrocannabinoid. So we know a lot about this in the media and politics, but as a natural chemical, people saying it helps, it's medicinal. Maybe less accidents with marijuana than there is with alcohol, but we seem to villainize people who've done marijuana. But the issue of how it functions, your body has its own marijuana receptors. Anandamide, or 2-AG. These receptors are responding to marijuana. It's like we say, you have your body's own endorphins or mechanisms that respond to deal with pain. You have natural elements that are in our universe, and your body has mechanisms to absorb those mechanisms. So the fact that you have natural mechanisms in your body to absorb marijuana, hey, maybe it's there for a purpose. But I'm not encouraging you to do that. So, as we close out on this discussion on synapses, there's some synapses that are very quick acting, Those areas that are melanated, darker in color, quick acting, fast, because the melanin helps to absorb energy, as in darkness. There's also something called gap junctions, where the receptors line up on top of one another and cause for easy flow. And those places are called gap junctions that have direct contact between the neuron membrane. This is important because this quick acting function happens a lot in embryonic development, where the brain is constantly developing quick and fast, 250 million neurons maybe a second as the child is growing and developing. So these gap junctions are very important for development. And again, also in the brain for quick acting, fast thinking. So a lot of areas that are actually melanated in the brain also have gap junctions. And showing the lining up, there's a presynaptic terminal, postsynaptic terminal. You see the actual channels are lined up. Makes for easy access for electrical activity. Hormones can also be labeled as neurotransmitters, but hormones are considered different because they are chemicals that are secreted by a gland. Could be the thyroid gland, could be the pancreas, could be the pineal gland, that transport it to other organs to have an effect somewhere else, to alter some activity in the body. So a chemical release from an organ to attack or to have an impact on another organ. So the collective structures that are emitting hormones are called the endocrine system. The endocrine system. Important for triggering, again, long-lasting changes. And we'll see some of these changes there in the chapter on reproduction. So here's some endocrine organs. Hypothalamus, pineal gland, pituitary, all in the brain. You have the thyroid glands, thymus gland, liver, adrenal, kidneys, pancreas, and then there's a chart that shows which each one of them actually releases. So, selected list of hormones and what they release. In the interest of time, we won't focus on that here. But as we conclude this, again, I said earlier, large chain amino acids are called proteins, small chain amino acids are called peptides. Chemicals that are released from the brain, you have the hypothalamus and pituitary. I'm not going to draw it because I'm going to show you another slide that has the images of where the hypothalamus is, and right below is the pituitary gland. The hypothalamus has releasing, it's releasing hormones, and the pituitary has your stimulating hormones. So they work in tandem. One talks to the other. in the posterior pituitary, that is really the glandular portion of the pituitary is really the first part, called the adenohypophysis. But just remember, it's the anterior pituitary. So look at the brain, front, inside, deep, there's a little triangle right there. You blow it up, that's the hypothalamus. There's multiple nuclei in the hypothalamus. Lateral, ventromedial, these multiple nuclei have different roles to play in terms of impacting the brain and behavior. So the hypothalamus then talks to the pituitary below, and you usually think of the pituitary as the master gland. So here you have bone. That blue there is bone. So the pituitary is actually sticking in bone in the brain. So the front part is where the gland, this part, backside, is really part of the brain. Posterior pituitary releases two neuropeptides, one called oxytocin, one called vasopressin. We'll see again later on, but write it down in your notes. There's two neuropeptides coming from the posterior pituitary gland. Next chapter, we're actually dealing with anatomy, and we'll see this a little deeper. Right now, we're just dealing with chemistry. So the hypothalamus, those multiple nuclei, all those little green structures of nuclei, they then come down to the pituitary gland, and all this blue is talking about the arterial system and how the chemicals are flowing there before they even go to somewhere in the body, make you grow, get bigger, affect your emotions. So these chemicals flow through here, go in and out. And so you got your stimulating hormones here, releasing hormones up here, and without that functioning, your whole body's out of whack. And again, it's showing that people with thyroid problems can have issues with being tired or metabolism issues, and so this slide here just shows you like a feedback loop to kind of cut things off, so there's not a whole lot of thyroid hormone being released, and can cause a problem if you don't have it being released. So this can be a very sensitive system. So in some, your hormones become then another potential target for helping people out with their thinking. Drugs could be focused on how those hormones work, and also we have to look at things like endocrine disruptors, chemicals that are in our environment that could impact the way these hormones work. So we're like a chemistry factory. So we sometimes mess it up by what we put into it and how we treat it. So we want to make sure we get the right chemicals in our bodies so our neurotransmitters and our hormones work properly. All right, so next chapter will be on anatomy.